What's up, Tumnad Lectures? In this video, we're going to talk about the proximal convoluted tubule. If it is your very first time to watch me, make sure you subscribe, you turn on a notification bell, so that whenever I load the video, you become notified and be the first to watch the video. So let's go ahead and get started and look at what we call the proximal convoluted tubule. The first thing to know when you're talking about the proximal convoluted tubule is to know what is proximal convoluted tubule and where is it going to be located. Okay, so now the proximal convoluted tubule is one of the structures that make up the nephron, and of course we said that the nephron is the basic structural unit of the kidney. Okay, so now the proximal convoluted tubule is one of the structures that make up the nephron. Okay, now where is the proximal convoluted tubule located? Because the kidney has what we call the renal cortex and then the renal medulla. So now all the proximal convoluted tubules, they are going to be located in what we call the renal cortex. Okay, now this is now a simplified structure of what we call the nephron. We are going to be having this first sac-like structure here. This is what we are calling the Bowman's capsule, and of course we're going to be having the glomerulus. Okay, then the first convoluted tubule just after what we call the Bowman's capsule is what we call the proximal convoluted tubule, and it's going to be the area of discussion in today's video. Okay, then immediately after the proximal convoluted tubule, we go to the thick descending loop of Henle, then the thin descending loop of Henle. Finally up moves what we call the thick ascending loop of Henle, and then this is the digital convoluted tubule, and then finally the collecting duct. Okay, but our main target in this video is going to be focusing on what we call the proximal convoluted tubule. Okay, now, during the discussion in the proximal convoluted tubule, we are going to be seeing what are some of those processes that are going to be happening in the proximal convoluted tubule. Mainly, we are going to be having the process of a tubular reabsorption, and of course, we're going to be having what we call tubular secretion. Okay, we are going to be having two processes. What are these two processes? We are going to be having what we call reabsorption. We are going to be having what we call reabsorption. Okay, and then we are going to be having what we call secretion. Okay, and because reabsorption in the proximal convoluted tubule, this is the tubule that has the highest percentage of substances get reabsorbed. Okay, so now, but of course you're going to be having some substances that are going to be secreted in the proximal convoluted tubule. But now we need to first understand what are these two terms mean. Now, any substance that moves from the proximal convoluted tubule or any of these tubules and it goes into the nearby blood vessels, it could be the peritubular capillaries. So long as it is moving away from the tubule and it goes into the medullary interstitium or it goes into the peritubular capillaries, that is what we call reabsorption. Things are getting reabsorbed back into the blood. Remember, they were once in blood, but because of net filtration pressure, they were filtered into the tubule, but they would wish to go back into the blood vessels. That is what is called reabsorption. But again, there are those substances which are going to get tired if they have stayed in the blood and they wish they want also come and test the sweetness of the tubule. Okay, these substances are moving from the nearby blood vessels and they come to test the sweetness of the proximal convoluted tubule. It could be even the distal convoluted tubule. It could be even the vasorector. It could be even any kind of area. So long as substances are moving from outside and they go into the tubule, this is what we call tubular secretion. Okay, this is what we call tubular secretion. We shall see what are some of those substances that get secreted. Okay, now these are the two processes which are going to be happening in what we call the proximal convoluted tubule. So now, following the net filtration pressure, I want to see what are those components that make up what we call the glomerular filtrate, because the substance which is going to be collected here in the Bowman's capsule is what we call the glomerular filtrate. But what are those components that are going to be accumulating in the proximal convoluted tubule that are going to be reabsorbing? Okay, now The most common of the substances that are going to accumulate here, we have what we call, the most common one, we have what we call water is going to get reabsorbed in the proximal convoluted tubule. We are going to be having what we call amino acids. Amino acids also going to be among the filtrate. We are going to be having what we call the glucose. We are going to be having what we call the lactate. We are going to be having what we call small proteins. We are going to be having what we call small proteins, and of course, smaller proteins are going to be having what we call the HB dimers and the insulin. We are going to be having electrolytes. Electrolytes like, for example, potassium, magnesium, calcium. We are going to be having even calcium. We're going to be having chloride. We are going to be having even what we call bicarbonate and a lot of substances. We are going to be having substances like urea. Mainly, these are some of the substances that are going to be contained in the filtrate, but they are bit very many, okay? But now we are going to be focusing and see how are these substances going to leave the proximal convoluted tubule and they go into the nearby blood vessels, which we are calling the peritubular capillaries. Now, there are three pathways through which these substances leave what we call the proximal convoluted tubule. What are these three ways through which the substances leave what we call the proximal convoluted tubule? So now I'm going to cut a small portion here. I zoom it here. We see exactly on a bigger interface and see what's going to be happening exactly. But before that, we need to first know what are the three pathways through which these substances are going to be leaving into the peritubular capillaries. Okay. Now, what is going to happen is that we have those substances that are going just to pass through the cell into the nearby blood vessels. They are going to be passing in the cell. They cross into the luminocyte, into the cytoplasm, and of course they cross outside the basolateral membrane into the blood vessels. So now, how do we call that kind of transport whereby the substances are going to be passing through the cell? This is what we call transcellular. Okay, this is going to be what we call transcellular pathways. Okay, and what are those of the examples that are going to be passing through this transport mechanism? We have what we call sodium ions. We have what we call glucose. We have what we call the amino acids. We have what we call the lactate, and of course we have what we call the lipids. Okay, the lipids. So also the lipids are also among the glomerular filtrates. Okay, now that is our first pathway. Okay, so now what is the second pathway? Now we have some of those substances that do not pass through what we call the transcellular mechanism between the cell, but they would love to pass through what we call the tight junctions in between the cells. Okay, they like to pass through in between the cells. For them, they just want to pass here. Okay, in between the cells into the nearby blood vessels. How do we call that kind of transport mechanism? That kind of transport mechanism is called paracellular, is what we call paracellular transport mechanism. Okay, what are some of those examples of substances that are going to be passing through this mechanism? The best example, we are going to be having what we call chloride ions. We are going to be having what we call magnesium ions. We are going to be having what we call calcium ions. We are going to be having a lot of substances going to be absorbed in this mechanism. And of course, even a very small percentage of water can be leaking through what we call the paracellular mechanism. Then there is also another last mechanism through which some of the substances are going to be leaving through this tube into the nearby blood vessels. There are these substances that are going to be having their receptors located on the luminous side of the cells lining the proximal convoluted tubule. And then they get taken in and then they cross through the cell, but when they are in a vesicle, they are going to be endocytosed. Okay, but of course, this is not going to be endocytosis, but this kind of transport mechanism whereby the substances bind on their specific receptors and they get taken up inside. They are broken down by lysosome enzymes into individual amino acids, and then the amino acids diffuse out into the nearby blood vessels. This type of transport mechanism is what we call pinocytosis. Okay, pinocytosis. And the most common example of the substances that are going to be passing through this mechanism, we have what we call the small proteins, like insulin, and what we call the HB dimers. Okay, the HB dimers. These are some of the substances that are going to be reabsorbed in the process we are calling the pinocytosis. Okay. So now let's go ahead and get started and see how these substances are going to be reabsorbed back into the peritubular capillaries. Okay. Now, we are going to first reabsorb what we call the sodium. How is sodium going to get reabsorbed? Okay, this is something which is very super important. Now, what you should know is that on the basolateral membrane of this cell, we have this transporter. It is pumping out three sodium ions. It is pumping out three sodium ions, and it is bringing in two potassium ions. Okay, so now this is what we call sodium potassium epiesis pump. It is constantly pumping out three sodium ions into the blood and then pumps in two potassium ions into the cell. What's the significance of this? It means we are going to be having a reduced levels of sodium ions inside this cell. Okay, the levels of sodium ions are going to be reducing in this cell. This is going to create what we call the sodium gradient. In other words, the sodium levels in the cell is going to be less as compared to the levels of sodium ions in the proximal convoluted tubule. Okay, so now when this happens, we are going to be having this channel which is going to be located on the luminal side of this proximal convoluted tubule. This is going to start allowing in movement of sodium ions. Okay, the sodium ions are going to start flooding in through this mechanism. So now, this sodium is very, very important in a way that as it is getting reabsorbed, it has the tendency of pulling other minerals to go with. Now, here we are going to be having nutrients like, for example, glucose. We are going to be having nutrients like amino acids. We are going to be having nutrients like amino acids. We are going to be having nutrients like lactate or lactic acids. So now, this channel, what we call the sodium channel, now, as these sodium ions moves into this cell, what happens? Sodium is very important. It likes and pulls this glucose also to enter this channel. It also allows also the amino acid also to pass through, and also it can also pull the lactate ions. Okay, it can also pull the lactate or the lactic acid in. So now we are seeing sodium is pulling glucose, is pulling amino acids, and at the same time is also pulling what we call the lactate or lactic acids. But of course, there are going to be different channels whereby sodium is going to be sodium glucose transporter, okay, or importer. The moment they are going to be moving in the same direction. Okay, so now this is how glucose, amino acids, and sodium are going to be transported by the help of what we call the sodium ions. That is something which is super very, very important. Okay, so now we have reabsorbed glucose, we have reabsorbed amino acids, and we have reabsorbed what we call the lactate or lactic acids. Okay. Now, how about substances like, for example, magnesium ions, the calcium ions, and also the chloride ions? How are they going to get reabsorbed? We said that these ions, they are the easiest to get reabsorbed. Why am I saying this? For them, they don't pass through in between the cells. They are not taken up into the vesicles. For them, they like the in between the space in between the cells. Okay, this is where these ions are going to be passing. They are going to be looking for this space in between the cells, and they cross into the nearby blood vessels, and they're going to finally find into the blood. These are the easiest way of these ions, how they are going to get reabsorbed into the nearby blood vessels. They are the simplest to get reabsorbed, and the process for reabsorption of these electrolytes is what we call paracellular pathway mechanism. But of course, the chloride ions is also going to be using another channel. There is this channel here which pumps in it pumps in sodium ions, but as it is pumping in sodium ions, it can also pull the chloride ions. So now this becomes the sodium chloride importer. So this is again another mechanism through these chloride ions can also get reabsorbed back into the into the peritubular capillaries. But now we want to look at how is bicarbonate gets reabsorbed. Okay, how is bicarbonate gets reabsorbed? Bicarbonate is a bit sweet in the way, in the mechanism how it is going to get reabsorbed, but let's go into it and see how the bicarbonate ions get reabsorbed. Now, in blood, we have what we call CO2, or what we call the carbon dioxide. Now, this carbon dioxide from blood is going to come and enter into this cell. Okay, this is going to be our carbon dioxide. Now, when this carbon dioxide enters this cell, okay, when this carbon dioxide enters this cell, carbon dioxide is going to react with the water. Okay, it's going to react with the water, and what is going to be formed is what we call carbonic acid. Okay, is what we call carbonic acid. And of course, there is a process. This process is going to be under the influence of an enzyme called carbonic anhydrase. Okay, it's going to be under the influence of an enzyme called carbonic anhydrase. What is going to be happening when this carbonic acid is formed? Carbonic acid is a very weak acid that can quickly dissociate. Okay, it can quickly dissociate. It can dissociate into two components. What are these ones? It can dissociate into what we call the hydrogen ions, and it can also dissociate into what we call the bicarbonate ions. This is where something becomes very crazy. We are forming bicarbonate ions into the cell, but again we want to expel the bicarbonate ions. Okay, this is how things becomes very crazy. Okay, now this bicarbonate which has been formed inside this cell, for it starts and moves out. Okay, it starts and moves out. It goes back into what we call the nearby blood vessels. And of course, it's going to be now, if there's a bicarbonate ions, of course, this is even how the pH is going to be regulated by the kidneys, okay, or the acid-base balance. Okay, if this bicarbonate gets moved into what we call the nearby peritubular capillaries, depending on the levels of the pH, okay, or the acidity or alkalinity in blood. So this bicarbonate ends up going into the nearby blood vessels. But now we want to look at these proton ions, okay? Now, we are going to be having this transporter. We are going to be having this transporter on the luminous side of this cell. This transporter is going to be pushing in sodium ions. It's going to be pushing in sodium ions. But as it pushes sodium ions in, it now helps the proton to come out. Okay, it helps the protons to come out. So now this is sodium. This is going to be sodium hydrogen antiporter. Okay, this is going to be sodium hydrogen antiporter, meaning that it is these sodium ions that its entry aids in the exit, or what we call the secretion of hydrogen ions into the luminous side of the proximal convoluted tubule. Okay, now what is going to be happening? We have bicarbonate ions, we have the proton ions. What is going to be happening? These bicarbonate ions, they are going to react with the with what we call the formed protons. Okay, what's going to be happening that the bicarbonate, when they react with the hydrogen ions, we are going to be forming what we call carbonic acid. Now, carbonic acid, under the influence of an enzyme called carbonic hydrase, is going to be facilitating the process of what we call the formation of what we call the carbonic acid. Now, this carbonic acid which has been formed is going later to split into water and then also carbon dioxide gas. Even this water which is going to be formed is also going to be reabsorbed. Okay, it's going to be reabsorbed. How is water getting reabsorbed? In the luminous side of the proximal convoluted, we have what we call water channels, what we call the aquaporins 1. They are the one responsible for the reabsorption of water. Now, when sodium gets reabsorbed, glucose gets reabsorbed, these two ions, when they get reabsorbed, who follows? Sodium and glucose. You know, water likes an area which is salty. Okay, so now for example, if there is this transporter here, it is transporting in sodium ions and is also transporting in glucose. It means now water is going also to start following where this. So now, how do we call this kind of pooling of water towards the area which is a bit salty? Okay, this is what we call this kind of reabsorption of water following the salty area of the sodium and the glucose. This is what we call obligatory. This is what we call This is what we call obligatory water. It's what we call obligatory water reabsorption. Okay, this is what we call obligatory water reabsorption. Obligatory water reabsorption, this is the process whereby water follows or water is pulled by this concentration of solids which are leaving, which are getting reabsorbed back into the peritubular capillaries. Okay, we said it is called obligatory water reabsorption. Okay. Now, we want to look at how about lipids. Okay, how about lipids? For lipids, it is the very simplest way of how it's going to get reabsorbed. Okay, and of course, when we talk about the lipids, they are lipid bilayer. They are very friendly. Okay, so now these lipids, they are just going to cross the lipid bilayer and finally they find themselves into vessels. But we have some of those substances that are going to be friendly to the lipids. Okay, we have those substances which are going to be friendly to the lipids. For example, we have what we call urea. So urea is friendly to the lipids. It can also go together with what we call the lipids. Okay. So now that's generally how some of these ions are going to get reabsorbed. But again, we said that we are going to be having reabsorption in the proximal convoluted tube, but at what percentage? What percentage of the reabsorption is going to be happening here? Okay, so now sodium ions, they are going to reabsorb up to 65%. Okay, water is also going to reabsorb up to 65%. Okay, then the glucose is going to reabsorb approximately 100% under normal physiological process. Again, also amino acid, it's supposed to be reabsorbed 100% under normal physiological processes, and also the lactate. Okay, supposed to be reabsorbed 100%. Okay, these are the percentages, and of course, you're going to be having also the bicarbonate. This one is going to be approximately 90% and plus. Okay, going to reabsorbed into what we call the proximal convoluted tube. And of course, we are going to be having now a reabsorption also in the other areas of the nephron, like the descending loop of Henle, the ascending loop of Henle, the distal convoluted tube, and the collecting duct. More of the reabsorption and secretion are going to be happening. Okay, now we said we also have something like a secretion, okay, tubular secretion. What are some of the examples of those substances that are going to be leaving the peritubular capillaries and they come into the tubules? Okay, what are some of those examples? Example of those substances that are going to be entering what we call the proximal convoluted tube from the outside environment into the proximal convoluted tube. They bit quite many. Okay, the most common ones which you should know, you're going to be having what we call the protons, as we have seen how the protons was coming here. You're going to be seeing also the potassium ions. Now, how is the potassium ions getting secreted in this tube? You see, these sodium potassium ATPases, we said they are all over this basolateral membrane. Even the whole of this area, we are going to be having these pumps, okay? These pumps, they are throwing out 3 sodium ions and they are bringing in 2 potassium ions. Okay, all over the basolateral membrane of these cells, we have these pumps. They are the ones that are going to be keeping reduced levels of sodium ions in this cell, which is going to be enhancing the reabsorption process. That is something which is very important. Okay, these sodium potassium ATPases all over the basolateral membrane, they are going to be there. 3 sodium ions and then pumps in 2 potassium ions. Now, You realize now we are going to be pumping in potassium ions into this cell. But again, approximately 90% of potassium ions, they are located intracellularly. But we're again adding more of the potassium ions, meaning that you're going to be having a lot of potassium ions into the cell. Okay, now what happens on the basolateral membrane? On the basolateral membrane, we have these potassium channels. What happens that whenever the levels of potassium ions have become high, some of these potassium through these channels, they get secreted into the proximal convoluted tubule. That is how the potassium ions are going to be taken into the tube, which you are calling the tubular secretion. Okay, what are other substances that are going to be secreted? We have what we call creatinine. Okay, creatinine. Also, creatinine is going to be secreted into the tube. What are other substances? We have substances like the toxins. The toxins, also the drugs. The drugs are also going to be secreted into the tube. We also have substances like uric acid. So now these substances, instead for them, they are going to be leaving from the peritubular capillaries and they come into what we call the proximal convoluted tubule. That's why we say they are called undertubular secretions, okay? The protons, the potassium ions, the creatinine, the toxins, the drugs, uric acid, all these substances are going to get secreted into the proximal convoluted tubule, okay? So having looked at that, we still have some of the substances which has not yet been reabsorbed. What are those substances? Uh, we said we have what we call small proteins. How are the small proteins going to get reabsorbed? Now, here is going to be my cell. This is going to be my cell. We said the small proteins, we have an example of what we call the hemoglobin dimers, and also have what we call insulin. Okay, so now how are these substances going to get reabsorbed? Now, on the luminous side of these cells, you're going to be having what we call receptors for these HB dimers, and also receptors of what we call the insulin. Okay, what happens that insulin will come and bind on these receptors and the HB dimers. What happens is that we're going to be having an invagination. Okay, we're going to be having an invagination. Okay, we are going to be having an invagination. Now, this invagination is going to be taking in all these substances that are bound into these receptors. And then finally, these substances are going to be more of endocytosed. Okay, they are going to be endocytosed inside here. Okay, they are going to be endocytosed. They are going to be endocytosed inside here. In this vesicle, you're going to be containing the hemoglobin dimers and then the insulin. Okay, so now we are going to be having what we call lysosomal enzymes, which are going to be coming and they start destroying all of these proteins, of these small proteins, into their individual. They are going to be broken down into their individual amino acids. And of course, these individual amino acids, they are the one which are going to live through the basolateral membrane and they get taken back into the nearby blood vessels. Okay, we said this kind of transport pathway through which substances are going to first get slightly endocytosed to make a vesicle inside and then broken down by the lysosomes inside these cells lining the proximal convoluted tubule into individual amino acids, and then the amino acids, they are going to go taken up into the nearby blood vessels. We said that kind of transport pathway, we said it is what we call pinocytosis. And we said the example of substances that are going to be reabsorbed in that way, we have insulin and also have what we call the HB and the HB dimers. Okay, that's generally what's going to be happening in what we call proximal convoluted tubule. We have just talked about that we're going to be having tubular reabsorption and also secretion. We have said that there are three pathways through which substances are going to be leaving the proximal convoluted tubule. We said in the first one as that transcellular substances are going to be passing through the cell, through the cytoplasm and into the nearby blood vessels. We called that one as the intracellular pathway, or what we call the transcellular pathway or transmembrane pathway. Okay, then we said there are some substances that for them they like passing through the tight junctions in between the cells. Okay, we said that these substances are moving out by the process called paracellular, and we said substances like magnesium, calcium, chloride, and very many others, they can pass through in between the cells. Okay, and then finally we said that there are some substances that are going to be binding to the specific receptors on the luminal side of these of these cells lining the proximal convoluted tubule, and they get invaginated in, forming a vesicle that's going to get broken down by the lysosome enzymes into individual amino acids, and of course the amino acids move out into the nearby peritubular capillaries, and we said that kind of reabsorption is what we call pinocytosis. Okay, and again we went ahead and said the example of substances that are going to be reabsorbed in this format is what we call insulin, and then the HB dimers among the small proteins. Okay, and then finally we said that you're going to be having tubular secretion, substances moving from outside into the tube. We said those substances, we have the likes of protons, the potassium ions, the creatinine, the toxins, the drugs, the uric acids, and a lot of substances. They are going to be secreted into the tubules. Okay? So now that is the brief summary of what we call the proximal convoluted tubule. If you find whatever I've just talked today is helpful, is adding to you, please make sure you subscribe, and you can share the video to the friend that you think it is going to be helpful, and don't forget to subscribe. You people your presence is always my happiness. As always, Tumunad, until we meet next time.